So one of the things that's changing rapidly in oncology is that the tumor, the tissue itself, taken out during a surgery, is becoming a critical component to making potentially life-saving vaccines for these patients. And so we're continuing to work with the surgeons to evangelize them as to the availability of these new vaccines so they can start including that into what they discuss with the patients.
When I see a patient with an advanced cancer, the role of immunotherapy, vaccine therapy opens incredible doors. So I'm thinking of every patient, of every tumor type, that this is a tumor that can be harvested, a portion of it can be removed, some of it can be sent for developing a vaccine. The rationale for debulking and removing tumor is entirely justified and appropriate in a circumstance where you have additional therapies that open tremendous doors to the patient.
We have many tumors where we have primary therapies that we can start out with, but there's some tumors that I think that the primary therapy is not particularly good, may be very toxic to somebody, and I think that's a person right there that can go on to a research study right from the beginning, or someone who's gone through one or two maneuvers and their disease is not under control and they still want to have additional therapy. Mary Crowley is also doing a lot of vaccine work, is doing a lot of gene therapy type work. They offer a therapy for my patients that they can't easily get in other places. It's one of the few ways that you can get these drugs. I mean, if you don't work with a group such as them, you're not going to be able to have the drugs for your patients. Now, all the drugs don't work, but it's much better than what it was before we had the targets.
The Mary Crowley tends to go from the laboratory and animal-based experiments and cell lines into people pretty quickly, feeling that it's very likely that it's safe and they will do the safety studies, but get it into the efficacy stages much more quickly. So we're collaborating on a particular study where women with metastatic breast cancer that have very highly aggressive disease that's already metastasized, their tissue has been harvested and a complete genome sequencing has been done. So I'm very, very interested in that because it really hasn't been done in this comprehensive way that Mary Crowley is doing it. I think the Mary Crowley's approaches are pretty incredible, very cutting edge, very, very highly innovative that no one else is really doing in the world. And so it's an opportunity to get access to a new therapy that you don't otherwise have access to, and to hopefully help yourself, and also, of course, to contribute to the larger body of knowledge, which is going to change the standard of care for the women's daughters.
Everything in the investigatory area of oncology is focused on not the sort of sledgehammer, smash the tumor kind of approach, but more the targeted therapies, kind of the lock and key approach. That means being able to evaluate the genetic and protein basis of the tumor and being able to differentiate that tumor from the normal tissue, and then selecting those differences to focus on an attack that allows you to kill the tumor cells preferentially over the normal cells. The type of therapies that are offered through Mary Crowley and the type of therapies that are offered in early phase studies in Texas Oncology are complementary. I think long term, you're going to see every single tumor will be sequenced and genetically analyzed as a standard part of the pathology evaluation.
Molecular biology and targeted therapy is revolutionizing and changing the way that we're treating cancer. What Mary Crowley has given is that hope that, the possibility that something new and something can turn the lives around. And just that, given that hope is such an important part on the healing process too.
I think the thing that's most exciting about working at this center, we really are customizing cancer care. So we are learning about the patient at a genetic level, at a DNA level, and identifying what is going to be the best treatment for that patient based on that, and then custom making treatments for the patient.
When the surgeon is removing the cancer, he has an opportunity in working with our program to actually provide us a small piece of the cancer. We'll have our technician in the operating room, or we'll train his staff, and we're developing kits that'll preserve the tissue so it can be used to make a vaccine and can be used to do the molecular analyses that could lead us towards therapies of the future.
But we did learn about the size of tumor that we need to get the current best vaccine. As of now, it's about a golf ball size piece of tumor that gives us the perfect size. The other thing we learned is that tumor viability is still important. So if we're ablating tumors, it doesn't help for us to nearly burn a tumor and to destroy it. And then finally, it doesn't make sense to give them high-dose chemotherapy and 2 weeks later expect to get a good viable tumor that we can turn into a vaccine. In general, though, the ease of harvesting a tumor is as simple as taking the tumor out, turning and handing it into a Petri dish where there's a waiting research assistant that's going to take that tumor, prepare it, send it to the lab, and roughly 6 to 8 weeks later, depending on the protocol or the sequence for that particular patient, they might be eligible to begin getting those subcutaneous injections. Having a collaborative relationship with Mary Crowley as a surgical oncologist is providing the absolute best care that you can to your patients.